Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Abah Iskandar bin Luqman. I'm from FPA200 3B. Today I'm going to present about bacterial heart rot disease. So for introduction, bacterial heart rot disease (BHR) is a disease that affects pineapple plants. It's caused by pathogenic bacteria such as Erdichiaceae or Erwinia chrysanthemi. BHR can cause soft rot in the fruit and blister-like lesions on the leaf. Bacterial heart rot is a destructive plant disease affecting a variety of crops, particularly those in the monocot family such as maize, onion and certain tropical fruit. So for the transmission agent for this disease is water. The contaminated irrigation water is a major vector for spreading bacteria. Overhead irrigation can splash bacteria onto healthy plant, leading to infection. Other than that, certain insects such as beetle, aphid or thrips can carry bacteria on their bodies or in their mouthpart. These insects can create wounds on plants while feeding, providing entry points for bacteria. Other than that, mechanical equipment. Some tools and machinery used for planting, pruning or harvesting can become contaminated with bacteria and transfer them from infected to healthy plant. And lastly, plant debris and soil. Bacteria can survive in plant debris left in the field after harvest. These residues act as a reservoir, allowing the bacteria to infect new crops in subsequent growing season. So for the symptom of this disease, firstly, soft water soaked lesions. The disease typically starts with small water soaked spots on leaf, stem or fruit. These lesions rapidly expand, becoming larger and more noticeable. Other than that, internal decay. As the bacteria invade the vascular tissues, the internal part of the plant, particularly the heart or the central portion, begin to rot. This results in a mushy, discolored interior, often brown or black in color. Next, cavity formation. Over time, the decayed tissue may break down completely, leaving hollow cavities within the plant structure. Other than that, foul odor. The rotting tissue often emits a characteristic of foul smell due to decomposition process, which can help in identifying the disease. And lastly, wilting and yellowing. In advanced stages, the plant may show wilting and yellowing of leaf due to disrupted nutrient and water flow. So for diagnosis of this bacteria, firstly, visual inspection. Initial diagnosis can often be made by examining the characteristic symptoms such as internal decay or foul odor. Next, lab testing. For a more accurate diagnosis, sample can be taken to a laboratory for bacterial isolation and identification. Techniques like culturing the bacteria on selective media, polymerase chain reaction (PCR) or serological can confirm the specific pathogen. The next one, the microscopic examination. In some cases, microscopic examination of the affected tissue may reveal bacterial colonies or specific structure like slime or ooze. And lastly, field test. Quick diagnosis tests such as tissue blotting or dipstick test can be used in the field or for rapid identification for plant infection. So for the disease cycle, firstly, the bacteria responsible for bacterial heart rot disease can survive in the soil, plant debris, or infected plant tissue between growing seasons. They may also persist in water source and on weeds, serving as reservoirs for future infection. Next, the bacteria typically enter the plants through natural openings such as stomata, lenticels, or wounds caused by mechanical damage, insect feeding, or pruning. Next, once inside the plant, the bacteria colonize the vascular tissue, particularly the xylem. They multiply rapidly and produce enzymes that degrade the plant cell walls, leading to tissue maceration and rot. Next, as the bacteria spread within the plant, they cause the characteristic symptom of bacterial heart rot, including water-soaked lesions, internal decay, and cavity formation. The production of bacterial slime or ooze is common, which can further aid the spread of the bacteria. Next, the bacteria are disseminated to new plants through various means such as water, insect, or human. And lastly, the secondary infection can occur when environmental conditions such as high humidity and warm temperatures are favorable. This leads to rapid disease progression and spread within the crops. So for environmental condition favoring the spread of the disease, firstly, the humidity and the temperature. The warm between 20 and 30 Celsius and humid conditions are ideal for bacterial proliferation and spread. High moisture level, especially in dense plant canopies, create an environment conducive to infection. Next, the poor drainage. Waterlogged soil or poor drainage system can facilitate bacterial survival and increase the risk of infection. And lastly, inadequate sanitation. The leftover plant debris, poor field hygiene, and lack of crop rotation can contribute to the persistence and spread of the pathogen. So for the impact on the agriculture, first, the yield loss. The disease can lead to severe yield losses due to the destruction of the plant tissues, particularly the heart or central part of the plant. Infected plants often fail to develop properly, resulting in lower overall production. Next, on environmental impact, the need to control bacterial heart rot disease leads to the increase of use of bactericides and other chemicals, which can have environmental consequences such as soil degradation, water contamination, and impact on non-target organisms. Other than that, economic losses. Farmers incur direct financial losses due to the reduced yield, lower market prices, and increased costs for disease management. For example, purchasing bactericides and implementing control measures. And lastly, the impact on trade and export. The severe outbreaks of bacterial heart rot can lead to export recession, especially if the importing countries have stringent phytosanitary standards. This can limit market access for affected regions. So for the management and control strategies, firstly, environmental management. The climate control in greenhouses. In controlled environment like greenhouses, managing temperature and humidity can help prevent the condition that favor bacterial growth. Other than that, use of resistant varieties. Planting resistant or tolerant varieties can significantly reduce the incidence or severity of bacterial heart rot. Breeding programs continue to develop new resistant strains to combat evolving bacterial pathogens. Other than that, cultural practices. So for crop rotation, rotating crops with non-host species help reduce bacterial population in the soil and minimize the risk of infection in subsequent planting season. Other than that, sanitation. Removing and properly disposing of infected plant debris prevent the bacteria from persisting in the field. Cleaning tools and equipment after use in the affected areas also help prevent the spread. And lastly, chemical control. The bactericides. The copper-based bactericides and other chemical treatment can be used to manage bacterial heart rot, especially during early stages of the disease or under conditions favorable for bacterial growth. However, over-reliance on chemical control can lead to development of bacterial resistance. In conclusion, bacterial heart rot disease is a significant plant disease that poses serious challenge to agricultural production, particularly in crops like maize, onion, and tropical fruit. The disease caused by various pathogenic bacteria such as Pectobacterium, Dicea, and Pseudomonas leads to internal decay, reduced yield, and economic losses. Effective management of bacterial heart rot requires an integrated approach combining cultural practices, the use of resistant varieties, chemical control, and biological methods. So that's all from me. Thank you.